Fermentation reactions of bacteria are valuable criteria for identification because not all organisms are capable of fermentation, and those that do can be identified by the sugar they are capable of metabolizing. Fermentation tubes contain peptone broth, phenol red indicator, and a sugar, such as glucose, sucrose, mannitol, or lactose. The fermentation test tube contains an inverted Durham tube, which also contains broth and is used as an indicator of gas production during the fermentation process by observing for trapped gas in the Durham tube. Be sure there's no airspace in the Durham tube. Different fermentable carbohydrates can be added to the broth to determine the fermentation reactions of a bacterial species. We will use fermentation tubes with sucrose and xylitol for the sources of carbohydrate. Label each fermentation tube. Use a loop to inoculate fermentation tubes with the bacterium to be tested. Flame the loop until it is red hot and let it cool. Uncap the tube containing the bacteria and pass the tube through the flame. Obtain bacteria using your loop and pass the tube back through the flame before recapping. Inoculate the first fermentation tube with the bacteria. Flame your loop and let it cool. Obtain an inoculum of the second bacterium to inoculate the second fermentation tube. After you have inoculated both tubes, reflame your loop. Turn off the Bunsen burner and incubate the tubes at 35 to 37 degrees Celsius for 24 hours. After 24 hours, observe your results. The indicator dye, phenol red, is red at pH 7. Fermentation that results in acid production will lower the pH and turn the indicator dye yellow. Gas produced during fermentation will be trapped in the Durham tube and will appear as a bubble in the upper third of the tube. Tubes incubated for longer than 24 hours may not give accurate results. These tubes were incubated for 48 hours. When the bacteria run out of carbohydrate, they use peptone, and the resulting ammonium ion increases the pH, changing the color of the indicator. If the tube remains red, gently swirl it from side to side to check for cloudiness of bacterial growth that may have settled to the bottom. It is possible that the culture did not grow, leaving the tube red, and providing a false negative result. Turbidity in a red tube, however, confirms a true negative result. Remove your labels and dispose of the tubes properly.